who is one of our past trainees. He initially studied biochemistry at McGill before moving to Vancouver to do his MD PhD at UBC. His PhD was under the supervision of Dr. Connie Eves, who's an internationally renowned stem cell biologist. And during his PhD, he was supported by a Vanier Canada graduate scholarship. He worked closely with BC Genome Sciences Centre to develop cellular barcoding as a high throughput and quantitative clonal analysis method for primary human cells. After his MD PhD, he did his training in medicine and medical oncology here at U of T. He moved early last year to Cambridge, where he's a clinical research associate at the University of Cambridge, working under the supervision of Dr. Carlos Caldas, who's a clinician scientist and a leading expert in functional breast cancer genomics. His fellowship in the UK is supported by both an ESMO Translational Research Fellowship and an ASCO Young Investigator Award, and he's going to present to us today on his current research. So take it away, Long.
Great, thank you so much, Andrea. And thank you for the opportunity to be here to share some of my work with you. Before I jump into my talk, I just thought I'd paint a bit of a picture of my research journey. So this was the very first lab that I joined with Callie Gehring in Montreal. And in his lab, we used nuclear magnetic resonance and X-ray crystallography to study the structure of different proteins and enzymes. And then in Connie's lab, I did my MD PhD. And for those of you who don't know Connie, she's a phenomenal mentor and a very well-recognized stem cell biologist. And she, in fact, just recently got the Order of Canada. So it's been a privilege to work as part of her group. And then I came back to Toronto, which is where I grew up. And this is where I did my residency training. And this is a picture of the group of us before COVID started. So it was actually very nice to get to know people before wearing masks and all of the restrictions. And as you can see, in Cambridge, in the UK, COVID restrictions are much more relaxed. So we were able to get together as a big group. And this is in the lab of Carlos Caldas. He's a clinician scientist in breast cancer and an expert in breast functional genomics. So I don't have any disclosures. And this is an outline of my talk. So I'll start by giving a bit of background, and then I'll jump right into a bit of technical details around the development of the cellular barcoding technology. And I'll be presenting version 2.0, which is a new and improved version and constitutes some of the work that I've been doing in the first few months of my fellowship in Cambridge. And then I'll backtrack a little bit, and I'll tell you about how we use barcoding to study normal mammary stem cell biology, as well as a model of de novo human breast tumor genesis. And then I'll tell you a bit about some of our preliminary work in different models of patient-derived xenografts. So we know that there are significant differences between breast cancers, and histologically, they can be divided into triple negative, ER positive, HER2 positive, with there being some overlap between ER and HER2. This has traditionally informed our treatments clinically. And then there was gene expression profiling that led to the development of PAM50 subtypes. And these included luminal A, luminal B, HER2 positive, and basal subtypes. And they showed differences in survival outcomes. They also show differences in their likelihood to metastasize to distant sites. This slide is from the Metabrick study, and this study was co-led by my fellowship supervisor, Carlos. In this study, they examined the genomic and transcriptomic profile of 2,000 human breast tumors. They found that gene expression in breast cancer is mostly driven by copy number alterations. And so by integrating copy number profiles as well as gene expression profiles, they were able to identify 10 novel integrative subtypes. These subtypes have distinct copy number profiles, which is shown here, and they also show distinct survival outcomes. And I highlight some of these subtypes. So there's integrative cluster 2, which is a high-risk ER-positive subtype and defined by its 11q13.14 cis-acting alteration. And this causes increase in expression of cyclin D1 implicated in cell cycle and developmental processes. There's also integrative cluster 10 that is a basal subtype and is characterized by a chromosome 5 transacting deletion. And then you have integrative cluster 4, which is characterized by its relative lack of copy number alterations. But besides survival outcomes, these subtypes can also be used to predict disease recurrence after an initial period of 5 or 10 years of being disease-free. So for example, I mentioned that integrative cluster 2 is one of the most high-risk ER-positive subtypes. And you can see that after a period of 5 years of being disease-free, the risk of relapse can increase to over 50% at 15 to 20 years. And then there's the triple negative subtypes, so integrative cluster 10 and 4 ER minus. You can see that there's a big difference between the patients in integrative cluster 10 that are disease-free after 5 years. Their risk of late relapse is pretty low compared to 4 ER minus, which is significantly higher. So looking at all of this information about breast tumor heterogeneity, one of the questions I had early on was, where does all of this heterogeneity come from? And that leads me to some of the work that was done in Connie's lab, because her mammary group was focused on studying normal mammary stem cell biology with the idea that understanding normal biology would give us insight into where this heterogeneity in breast cancer is derived from. The schematic here shows a cross-section through a mammary duct with the inner cells being luminal cells, and these are surrounded by a layer of myoepithelial or basal cells. And early work in her lab before I joined showed that it's in the basal fraction of cells where you find the most mammary repopulating units, or MRU, and these are the functional equivalent of long-term, sorry?
Sorry.
Yeah, so
Sorry, I was looking actually a place to watch the meeting. Sorry.
You're welcome to join me if you
Yeah,
want.
no, actually, I have to prepare for the interview. It's like
Can you guys mute your line? Thanks. Keep going along. Sorry about that.
Yeah, no
We're joining
problem.
from all over today, so thanks.
In her lab before I joined, they showed that it's in the basal cells that you have most of the mammary repopulating unit activity. And these are the functional equivalent of long-term regenerating cells or mammary stem cells. So this work allowed them to construct this conceptual framework of the mammary stem cell hierarchy. So the mammary stem cell resides at the top, has self-renewal activity, as well as long-term regenerative activity. And then in the middle, you have these progenitors that have limited regenerative activity and can form colonies in vitro. And then at the bottom, you have terminally differentiated cells with very little or no regenerative activity. On the right here is a depiction of this cancer stem cell concept. And it's an updated view. The cancer stem cells are in red, and they can be influenced in terms of what regenerative activities they display based on a series of extrinsic factors such as niche signals and the tumor microenvironment. And there may also be an element of interconversion seen between cancer stem cell and non-cancer stem cell states. So when I joined the lab and started my PhD, my first research question was, what is the heterogeneity of regenerative activity in normal mammary cells? To address this question, we needed a clonal tracking strategy, such as cellular barcoding. But as I mentioned, I'm going to be talking about the second generation of barcoding, which is what I've been working on at the start of my fellowship. So for our barcoding strategy, we use the P-Larry vector that we received from the Camargo lab in Boston. It's driven by an EF1 alpha promoter that drives the expression of GFP. And downstream of GFP in this 3' untranslated region, is a cloning site where we inserted our barcode. And the advantage of this is that when GFP is expressed into mRNA transcripts, so is our barcode. And this makes it compatible with single-cell RNA sequencing. Our barcode design is fairly simple. It's 31 base pairs. And at the beginning, there are 4 nucleotides that denote a library ID. And this allows us to generate multiple different libraries with the same barcode structure. The barcode structure itself have these N's and S, which are the variable regions, and they're interspersed with constant sequences that provide structural integrity. And with this design, we can have over 4 million unique barcode sequences. On the left here is how the barcoding method works. So we have the barcode plasmids that we package into lentiviruses that are used to infect our cells of interest. And eventually the cells will then express GFP. We can assay the cells in whatever experiment we want. And at the end, we retrieve the cells and extract DNA, amplify out the barcode and sequence it. And then there are also several downstream analysis steps as well, some of which I'll get into later. But the idea here is that the initial cells that we put in are individually marked. So each cell has a unique barcode. And then at the end, when we sequence it, we can see what those cells at the beginning were able to produce in terms of clonal regenerative activity. But one of the first important technical things we had to do was to sequence our plasmid library to ensure that we had a decent diversity of barcode sequences. So the plots here show the first barcode library and the second barcode library, and each dot is one of the unique barcode sequences. And on the y-axis, the log of the number of reads when we sequence them. And you can see that there are a handful of barcode sequences that may be a bit overrepresented, but the majority of the barcodes are fairly evenly represented. But importantly, we found that in our libraries, we had between 170 to 220,000 unique barcode sequences. And that was pretty good for our purposes. The other technical control we had to do was we needed to see how much of the plasmid diversity is captured in our infected cells. So for the 2 libraries, we infected cells in triplicate, and we sequenced them to see what barcodes could be detected. And we found that between 30 to 35% of the plasmid library is captured in our infected cells. And this is pretty good compared to some other groups that have done this in the literature. The other thing with this method is we wanted to measure the sensitivity of clone detection, because that's pretty important for our purposes. So to do this, we established 6 different controls. So these are cell lines with known barcodes that are spiked into our sample at known cell numbers. So when we sequence them, we expect to have these barcodes only and not any other aberrant barcodes. And we showed that using this method, we could detect clones that were 50 cells in size 100% of the time, and clones that were 10 cells in size 90% of the time. But with this method, we're also able to construct this relationship between the number of reads. So here the FRV is a fractional read value. So it's a normalized read count between different multiplex samples, as well as the input cell number that we know for our experimental clones, for our controls here. Using this relationship, we were able to define what's here in gray, which is this threshold. So above the threshold, we can be very confident that all of these are real clones. But below the threshold, we get into the area of a lot of noise and sequencing error. And so traditionally in our previous method, when we apply the threshold, we basically lose all of these small clones. But with our new approach, we actually compare the clones below threshold to a whitelist of previously confirmed barcode clones that we establish by sequencing our plasmid library. And by doing this, we can improve clone detection to a sensitivity of 90% for these small clones. So to just conclude the first part, we're able to increase the sensitivity of detecting very small clones that are 10 cells or less in size by comparing back to our whitelist of known barcodes. And I constructed 2 libraries that we have validated, and this will be important for later experiments that I'll tell you about. And it allows us to do sequential barcoding experiments that will investigate subclonal activity. And this barcoding approach can be used for any mouse or human cell type, particularly cancer cell lines or PDX models. So now moving on to work that I did in Connie's lab. So this work I did in very close collaboration with Maysam Makram, who's currently a 5th-year resident in the Toronto Medical Oncology program. So I know a few of you will know her. So the experimental design that we came up with was to look at these basal human mammary cells, because within this population of cells is where we identified most of the mammary stem cells. And so we barcoded these cells, we transplanted them into our in vivo assay. After 4 weeks, we got mammary structures that we then dissociated into a single cell suspension, sorted into luminal and basal cell types, and then sequenced them. We then retransplanted some of the basal cells as well into secondary mice and looked at what they produced and did the same thing. But unfortunately, what we found in the human model was that the clones were very small. So in the primary mice, they were less than 100 cells, and in the secondary mice, they were a bit larger. But this wasn't really enough for us to decipher the range of regenerative activity from mammary cells. So then we decided to look at the mouse model for the mammary gland. I show here the clonal outputs from the primary mice and the secondary mice. So in purple here are the bilineage clones, so clones that produce cells of both luminal and basal types. And then you have the lineage restricted clones. And on the scatter plot here, each dot represents an individual clone. And you can see that the bilineage clones seems to be the largest, but they have a relatively equal contribution from both luminal and basal lineages. The other thing that we can do with this is we can track the lineage output from these clones in primary mice and compare them to what they produced in secondary mice. So these clones here are the clones that were bilineage in the primary mice, and we look at their output in secondary. In gray are the clones that did not read out in secondary. So you can see that most of the bilineage clones retained their differentiation activity. So they produce mostly bilineage clones. But luminal clones produced half luminal and half bilineage clones, suggesting that lineage differentiation patterns are not necessarily preserved upon serial transplantation. So to conclude this part of the talk and summarize the types of clones that we detected, we showed that there were short-term clones that do not propagate in secondaries. Then there are long-term clones that don't really maintain their size, so they get smaller in secondaries. There are also clones that are very robust, so either they maintain or expand in size in secondaries. And then there are also delayed clones. So clones that are not initially detected, but then read out in secondaries, suggesting that they're initially dormant. So going back to this slide, we now show that there's a lot of heterogeneity in regenerative activity from normal mammary cells. Now, I wanted to know how the cell of origin contributed to the heterogeneity that we observe in human breast cancers. So to address this, we wanted to develop a model of de novo human breast tumor genesis. So taking normal human mammary cells and transforming them into malignant tumors. And I'm borrowing this example from the colon, another epithelial tissue. And this is work from Bert Vogelstein and others. And because the dogma in the field at the time was that human carcinogenesis was a slow process, requiring the acquisition of multiple driver mutations over time. And so when I pitched the idea of generating malignant tumors from fresh human reduction mammoplasty samples, I was obviously met with a lot of skepticism. But in the end, we decided to go for it. And we showed that when we sorted luminal progenitor cells and basal cells, and we infected them with 3 oncogenes, KRAS, PI3 kinase, and a gain-of-function TP53 mutant, we could get tumors that formed within 8 weeks. in the end, we ended up showing that even just a single oncogene, this KRAS G12D mutant, was able to generate these tumors robustly within 8 weeks. And these tumors were serially transplantable, so they formed secondary tumors as well. So this system was very robust and could be done from 37 different patient samples. And the tumors formed under the kidney capsule of immunodeficient mice or subcutaneously. And when we look at the phenotype of these cells or of the tumors, we found that there was a reversal of phenotype, which is not what we expected. So both basal cells and luminal progenitor cells are normally ER negative. But when we transformed the basal cells, they acquired more luminal features and their tumors started to express higher levels of ER. So luminal markers that were expressed in these tumors were keratin 8 and 18, as well as MUC1. And in the luminal progenitor-derived tumors, you can see that they had much lower levels of estrogen receptor, as well as weaker staining for them, but they expressed both luminal markers and basal markers. So the basal markers here were keratin 5 and keratin 14. When we did gene expression analysis on the tumors, we found that they more closely resembled each other than they did the normal cells from which they were derived. And when we did PAM50 analysis, we found that they were more closely resembling normal-like breast cancers, suggesting to us that it was early in their transformation process, being only 8 weeks post oncogene overexpression. But the other unique thing about this model was that it allowed us to look at very early changes in these clonal populations during the process of tumorigenesis. So if we looked at if we look at 2 weeks before obvious tumors started to form, you can see that there's a small subset of clones that are already becoming more dominant in this clonal population. And then by 8 weeks, this is what it looks like. But when we looked at the tumors that formed just from KRAS alone, their clonal population already resembled that of the tumors at 8 weeks, suggesting that KRAS alone has a very dominant effect in advancing tumor progression. So to conclude this part, we found that within 8 weeks, just from a single oncogene, we could derive invasive and malignant human breast tumors that could be serially propagated. And we also found that there was a reversal of phenotype. So basal cells produced tumors with more luminal features, and luminal cells produced tumors with both basal and luminal features. And then by doing PAM50 analysis, we showed that the tumors were more normal-like, and to us, this suggested that we had captured them at an early stage in their transformation process. So moving on to some of the new work that I've been doing in Carlos's lab with different subtypes of breast cancer and PDX models. But before that, going back to this concept of the cancer stem cell concept, to me, the cancer stem cell concept suggests that not all cells have regenerative activity. So this plays into the concept of intra-tumor heterogeneity. But one of my questions was, how does intra-tumor heterogeneity differ between the different subtypes of breast cancer? And what are the molecular mechanisms that govern cancer stem cell activity, and how does this change with treatment? But to follow what I'll show you next, it's really important to understand the type of clonal information that we'll be looking at with cellular barcoding and the distinction between a genomic clone and a functional clone. So genomic clones are clones that are defined based on their unique mutational composition. So for example, with this ancestral clone, it acquired 2 separate mutations producing subclone A and B. Subclone B was genetically unstable, and so it acquired multiple different mutations leading to the development of subclone E. And you can see that by the 4th passage, subclone E had dominated and overtaken than the majority of the tumor. And if you look at the prevalence of these mutations within the tumor and construct these subclonal hierarchies, you can track their contribution to the overall tumor over time. A functional clone, as detected with cellular barcoding, looks at the regenerative activity that a cell can demonstrate independent of its genetic composition. So if you look at this population, we don't know, what mutations are in these cells, but some of them will show stem cell activity and some will not. So this is the idea that there are potentially non-genetic determinants of regenerative activity, such as extrinsic factors and epigenetic factors, because even if you had a population that was of the same genetic makeup, so all of the cells have the same mutational profile, some of them will be able to form tumors and some will not. But the power that we have now with single cell sequencing is that we can capture the regenerative activity with cellular barcoding and compare it to its gene expression profile with single cell RNA-seq, as well as its mutational profile, using single cell RNA-seq methods to do S&V calling and copy number profiling. But a more powerful way is to do single cell DNA sequencing with a method developed in Vancouver by Sam Aparicio called DLP+. So when we use our barcoding method and we look at different PDX models and we passage them serially into secondary and tertiary mice, we show that there are 5 distinct patterns of clonal growth. So clones that maintain their size, increase in size, decrease in size, and clones that die off, so only appear in the primary. And then there are these clones that are initially dormant, so we don't see them in primary mice because they're too small to be detected, but then they form relatively large clones in the secondary. So the dormant clones, to me, are interesting because it suggests that when the actively growing clones in the tumor are depleted, there's a reservoir of cells capable of taking over. And this is a concept that's been demonstrated in the normal colon epithelium with LGR5 positive colon stem cells. In one of these models, we were able to detect liver metastases. And when we analyzed the clones in the liver metastasis, we found that there were 2 types of clones here. So 52 of these clones were also detected in the primary tumor. But 140 of these clones were not detected in the primary tumor, suggesting that they didn't form large enough clones in the primary before they escaped to form metastases in the liver. And the clinical implication here is that a biopsy at one site is not necessarily representative of the clonal populations at the other site. To look at the differences in cancer stem cell activity between subtypes of breast cancer and how this changes with drug treatment, this is the experimental design that I came up with. So to start, we selected 4 different PDX models, 2 that are triple negative, 2 that are ER positive luminal B type, but they all belong to different integrative clusters. And we're in the process of expanding this to an additional 24 models. But how the experiment works is we dissociate these tumors, deplete them of mouth cells, and then barcode them with our first barcode. Once we get the tumors and we analyze them, we take a fraction of these cells and we barcode them again, but with a different library. So I mentioned this series sequential barcoding, and the importance of that is that we can look at subclonal structure. So for example, clones A, B, and C here are clones that we labeled with the primary barcode, and then clones 1, 2, 3, 4 to 6 are clones that we labeled with the secondary barcode. But we know that clones 5 and 6 came from clone C, clone 3 and 4 came from clone B, and so on. And so then when we do drug treatment, we can look at how these numbered clones respond to drug treatment. And if they respond in the same way, then their response to drug therapy is most likely intrinsically determined. And if they respond differently, then it's more likely that it's from extrinsic factors, such as the niche and tumor microenvironment, because within one passage, it's fairly unlikely that they would have acquired additional genetic mutations. But with single cell sequencing, with RNA and DNA, we would be able to tell if they did acquire additional mutations or not. So we've analyzed 3 of the models so far, and these are the total number of clones that we detected. We can calculate this clone initiating cell frequency, so the functional equivalent of a cancer stem cell, based on the number of clones we detect and the number of cells that we injected per mouse. So we can already see that between the 2 triple negative models, they have lower clone initiating cell frequency compared to the ER positive model that has a higher clone initiating cell frequency. We can also look at the prevalence or the size of these clones as a proportion of the entire tumor. And in the 2 triple negative models, we can see that even though there are fewer clones, there are only a handful of these clones that basically dominate the entire tumor. And in the ER positive model, there are many clones and they contribute relatively equal amounts to the tumor overall. The other thing that's interesting, and I didn't show this, are their tumor growth curves. They're fairly similar to each other. So it suggests that this tumor grew, in that period of time from the contribution of many clones, whereas this tumor was able to grow in the same period of time from just, 2 or 3 dominant clones. And the other thing that's interesting is I showed you that on serial transplantation that you get initially dormant clones. So clones that don't appear in the primary that then appear in secondaries. So I think in future experiments, it'll be interesting to see which of these clones continues to appear in subsequent passages. And I'm showing you here is the absolute clone size, so the distribution of absolute clone size. So in the triple negative models, you can see that there are very few clones that are very large. And then in the ER positive model, there are many more clones, but they're all fairly small. The other thing that we can do with this type of experiment is to analyze the clonal outputs of these PDX models by sorting cells based on their cell surface phenotype. And as a pilot experiment, I just looked at CD49F, and this is alpha-6 integrin. So what I'm showing you here is sorted for CD49F positive on the y-axis and CD49F negative on the x-axis, where each point represents a unique clone. The size of the dot represents the overall clone size, and the color is just the replicates that we did. So if the clone had equal contribution from the 2 cell types, they would lie on this dotted line. But the interesting thing that we found with this ER positive model is that there appear to be 2 kind of clusters of cells. So this cluster contains some large clones, but they have relatively equal contribution from the 2 cell types, whereas this cluster is predominantly CD49F positive. So it'll be interesting to see how these 2 clusters of cells respond differently to drug treatment. We also did an initial pilot run of single cell RNA sequencing on our barcoded tumors. And I'm showing here that we found 11 distinct cell clusters based on their gene expression profile. And all of these clusters are human cells because we depleted for mouse cells. So now what we're doing is we're analyzing the barcoding data from this. And we're extracting the barcode data from the unmapped reads from single cell RNA sequencing. And so the goal is then to look at, for the dominant barcode clones, where those cells lie within this plot, and how does that change with drug therapy. So we're currently undergoing the drug treatment trials with our mice right now. So I don't have that data to share with you, but stay tuned for this data in the near future. So to conclude this part, we showed that from PDX cells, we have 5 distinct patterns of clonal growth activity. In the metastatic clones, some of them form large clones in the primaries before metastasizing, and some do not. In the ER-positive tumors, we have more clones that contribute to a smaller extent, but equally to the tumor growth. Whereas in the triple-negative models, there are fewer clones, and a small number of them basically dominate the overall tumor growth. And lastly, we identified 11 distinct cell clusters in our barcoded tumors from single-cell RNA sequencing. So just to wrap up and kind of give an overview of the main points that I showed you from my work today. One of the main things is that we developed this barcoding version 2.0 that is compatible with single-cell RNA-seq, and we improved the sensitivity. So it's now very highly sensitive for detecting small clones. And because it can be coupled with single-cell sequencing, we can get genomic and transcriptomic information, meaning that how a clone behaves or responds to drug treatment can be linked to its gene expression profile, copy number landscape, and even SNV profiles. The other thing I showed you is that normal mammary cells display a broad range of regenerative activities, and actually that the cell of origin is not a good indicator of the resulting features of the malignant tumor. And one of the breakthrough findings that I showed you is that we were able to get malignant tumors to form from normal human mammary cells, fresh from reduction mammoplasty samples with just a single oncogene, KRAS G12D. And this model is useful for studying early changes during the process of tumorigenesis. I also showed you that PDX cells display 5 patterns of clonal growth, including some that are initially dormant. So these clones may be the ones responsible for relapse following treatment that depletes for the more actively growing clones. And not all clones responsible for metastases are actually detectable in the primary tumor, suggesting that a biopsy at one site may not be representative of the clonal composition at the other site. And in triple negative tumors, there appear to be fewer clones compared to ER-positive tumors, but the few clones that do grow out in the triple negative grow very aggressively and very quickly. And to me, this is in keeping with how we think of triple negative breast cancer in the clinic. And lastly, with single-cell RNA sequencing, we showed that there's 11 different cell clusters based on gene expression profiles, and stay tuned for how we link this to their barcode identity and how these change on drug treatment. So now I'd like to touch base a little bit in terms of my future directions. So my first aim is to establish a mouse hospital to efficiently derive PDX models that are treated in parallel to the patient. So the idea here is that for a particular patient, a biopsy sample before and after treatment will be used to establish PDX models. And these models can be used to compare the changes in clonality that occur with treatment. But the key thing here is that this resource is renewable because the PDX models can be expanded. So you can test many, many different drug treatments to assess their clonal changes from the original patient sample. And this is as opposed to a clinical trial where each patient can basically only receive one treatment at a time. So this is something that is being done in Cambridge, and their manuscript is currently in preparation. So it's proven to be feasible, but I know it is a very resource and labor-intensive investment. But I think it's something that would be very valuable if it were to be established in Canada, because it would allow us to look at clonal changes with treatment and identify specific drug-resistant mechanisms that can then be studied in much more detail. So what I would propose is to establish this with a small subset of patients as a proof of principle, and then slowly build the biobank over time. My second aim here is also related to the first, but to use barcoding and single-cell sequencing technologies on the different PDX models as we treat them and passage them to identify mechanisms of resistance that are specific to individual genomic clones. My third aim is to develop this functionalized barcoding strategy that can be applied to our PDX models. And the purpose of this is so that we can select for drug-resistant clones and then establish an organoid biobank of these drug-resistant clones. So the functionalized barcoding system has been established, and it's been proven to work in cancer cell lines for human breast cancer and human CLL, but it hasn't been applied to PDX models yet. But how the method works is you have these guide RNAs, but you generate the barcode library from the guide RNA sequence. So you have the unique sequence embedded within the guide RNA, and you use these to infect your cells. So you have a clonal population of cells all infected with unique barcodes. But then when you do your analysis and you find that there's a specific clone of interest that was either resistant to treatment or had a specific distinguishing a response in terms of the gene expression to treatment, and you wanted to study it in more detail, you can target those clones and select them by designing this recall plasmid that has a guide RNA recognizing site that is specific to that barcode sequence. So only those clones of interest will then express your reporter fluorescent gene, and in this case, it's GFP, and then you can select for these cells by flow cytometry. So the idea here is that you can gather a collection or a library of all of these interesting clones that have unique or particularly relevant responses to drug therapy, and you and you have this resource to examine these mechanisms in much more detail. And then from this biobank, you can do interesting things such as more high throughput drug screening to see if there's a particular drug combination that would be more effective than what was tried in the clinic. And you can also do CRISPR screening to identify genetic susceptibilities. And my last aim is to use all of the single cell gene expression data that we acquired through the previous aims to derive a single cell gene expression signature that can predict clonal response to therapy. So the example is if we have an initial pathology sample from a patient, we can send that for single cell RNA sequencing and identify the presence of clones that will likely respond or not respond to specific therapies so that we can tailor our first line treatment or even adjuvant treatment based on this information. Lastly, I'd just like to acknowledge everyone that was involved in the work that I showed you today. On the left are all of the people, mostly from Vancouver, in Connie and Sam Apricio and Martin Hurst's lab. And then on the right from Carlos's lab. And then my funding during my PhD, so the Vanier and CIHR and UBC, and funding during my fellowship, ESMO, ASCO Young Investigator Award, and CRUK. Yeah, CRUK. So